Oh, really, thank you for joining us. And
Well, it's my pleasure because this really is the future, and for your residents, it's going to be a reality at some point. So it's important to know about it. Um,
no, terrific, terrific. But I think the spectrum of knowledge and expertise that you have in so many areas is just amazing. I want to be like you when I grow up.
well,
Uh,
when you retire, quote unquote, you have to do something with yourself,
oh,
right?
amazing. Listen, I retired. I was a pen. I think you went to my old place, Pennsylvania Hospital, to give an Israel lecture, right? Some years ago, I think.
I'm sorry, where was that?
At Penn, at Pennsylvania Hospital, I think.
Yeah, and I will be there again. I think they may have given me a date, but I'd, s- I've certainly been in touch with them about a visit. So
And so Christos is one of my closest friends, Kutifaris. And
yes, same here. Same
yeah,
here.
so we were together for many years. I was a year ahead of him in training.
I see.
But I'm sorry, I'm not as smart as an REI. I'm just an MFM. So
Well, you should know that we have great respect for FFMs. Uh,
it's great. It's great. I saw that you went to med school to Tel Aviv University. My dad, that was a physician, he had made Aliyah, and he actually was in the faculty.
I see.
Sackler, in his later years, teaching in particular the Sackler, the program they had for the Americans.
Yes,
He was teaching there for many years until he died. So
I see. That's wonderful to know. I
yeah,
didn't.
my whole family is there, and actually, I lost a nephew on October 7th, a young doctor that worked at Soroka but lived in Berea. So
I see.
the the whole thing is a nightmare. So
I'm so sorry.
anyway,
Yeah, no, I mean, the things that come with aging, uh, including what you described, the loss of your father, the loss of a nephew, those are hard to deal with. And all of us obviously are contending with it in one fashion or another.
you're correct. You're correct. So I think we're gonna wait a few more minutes.
Sure,
We're supposed to start at 7:30, but
that's fine. We cleared up the time. We should be fine.
and I was looking. You were at Temple some time ago as a formal lecturer, right? I think you have given a talk maybe in Zoom a couple of years ago, but I think you were here actually visiting with Enrique, I think, sometime.
Yes. So, and Enrique, and I mean, I visited regularly at your place thanks to Enrique, whom we knew since he was either a resident or a fellow
At Hopkins,
at
probably.
Hopkins. Yes.
Yeah,
So that acquaintance goes back a long
many years.
time.
Yeah.
He wrote to me that he won't be able to be here today for medical reasons. But he's somebody I've been in touch with, and whenever I'm in Philadelphia, I make a point of meeting with him.
Oh, how nice.
Yeah.
How nice.
Now, it hasn't been a while because of COVID and some other issues, but as I said, I hope to be at Penn in person and possibly there. I'm not sure there's anything else coming up in Philadelphia at this point. I did speak at Jefferson not too long ago.
This is before Bill retired, or
No, long after he passed away, and
it was so sad. I was fortunate that I participated in his birthday party 3 weeks before, and it was just beautiful because all of probably your friends were there, who is who, and the
Yes,
talk of REI.
I
Yeah,
know.
it was very touching. It was unbelievable.
Yeah, I couldn't make it. Part of it, as I said, there were some medical issues at home that I needed to attend to. So, but yeah, Bill and I, again, we knew each other way back from Baltimore, and we were close friends. We co-authored papers. It was a blow, a major blow.
Very,
Um,
very, very fine.
so we're very cognizant of that. I watched if you can call it that, the ceremony at the synagogue.
Yeah,
They
yeah.
had it on
And
video.
so, yeah, we were there, and it
I
was touching.
imagine.
Yeah,
Yeah,
it was very touching. Very, very touching.
yeah,
Anyway, may his memory be a blessing. That's the only thing I can say.
very much so. And during the months prior to his passing, I was on the phone with him at least every week or two,
Really.
until at one point I couldn't make an appointment. And that's when Christos told me that he was an inpatient.
Yeah.
That was like a day or two before he passed away. But up to that point, we were in close contact.
Let me
And one of the reasons I'd like to be there in person is to be able to see his wife.
Lorraine, yeah.
Yeah, because it's not,
She has had a hard time. I've been in touch with her, but for
yeah,
obvious reasons. Plus, the kids are no longer here in Philly, so
I know. Well, but I'd just like to see her in person and say whatever I can say at that point. Um, so that was one of the reasons we wanted to make it an in-person grand round. Um,
well, let me just see. I think we'll wait one more minute
sure,
and I will introduce you. But again, thank you so much for
Jack, it's my pleasure, my distinct pleasure.
well, let's just start. Good morning, everyone. It is really my distinct pleasure and honor to introduce our invited lecturer that is not strange to Temple, Dr. Eli Adashi, that is a professor of medical science and the former dean of medicine and biological sciences at Brown University. And I hope that the residents in particular and the students are aware that what you're going to hear are the words of one of the true giants in the field. Dr. Dashi is an REI. He went to med school in Israel and then did training at Tufts and fellowship at Hopkins and REI and UCSD and was in the faculty in Maryland, and then for many years was the professor and chair of OBGYN at University of Utah before moving to Brown institution in which he has remained. He was dean for many years and actually currently is a tenured professor. And Dr. Adachi has been responsible for so many advances in the field of REI and trained so many mentors. And nationally and internationally, he's a member of the National Academy of Sciences and has been on the board of many institutions. the most amazing thing about Ellie, I'd have to say, is that every couple of weeks he sends a beautiful email to all his friends, quote unquote, and it starts like this: dear, in my case, dear Jack, here's hoping that all is well as can be, and please click here for a new paper. And the last 2 months, every couple of weeks, I get a new email from Ellie, and the spectrum of the topics that he publishes in is just amazing. The last one is about physician shortage. Prior to that, about Medicare. And one, I think, Ellie, like a month ago, discussing really giants in the field and the relationship between a Nobel laureate, Robert Guillemin and Sam Yen. So it is really an honor. And right now, Dr. Adachi is going to talk to us about something that is probably going to be the future, eggs and sperm from buccal smears. So, Ellie, I really want to thank you for, first of all your contributions to the field, for your mentorship, for teaching us that there is no limit in which someone could be, producing, collaborating, and in particular educating. So, without any further words, Ellie, please go ahead, and thanks again.
Well, thank you, Jack, and it's a pleasure to be at Temple again. I've been there many times pre-COVID, I should say, and in those days in person. And so I feel warmly towards the institution, and this is a revisit, albeit online. We will speak today about in vitro gametogenesis, which I think we can safely say is the next big thing. All of you, whether you were around in 1978 or not, are cognizant of the fact that perhaps the most significant breakthrough in our lifetime was the development and implementation of IVF, which began with the first birth occurring in 1978, the leading protagonist being Bob Edwards. That particular discovery, you may also be aware, was the recipient of a Nobel Prize in 2010. So by all accounts, particularly for reproductive endocrinologists, in vitro fertilization was and remains the most significant breakthrough in our lifetime. What we will be speaking about today, in my estimation, is going to be of a similar magnitude. It will impact IVF as well, but it will have many other implications which we could not have foreseen just a few years ago. And so what I'd like to dedicate our time today to is the notion of in vitro gametogenesis. It's a little hard to pinpoint the beginning of this field, but at the very least, we can go back to 2009 to a paper which was published in Cell by Japanese investigators who are still the main leaders of this field and who are making it happen. And so, for the purpose of our conversation today, we can assume that the beginning and the unfolding of this new development dates back to 2009 and possibly prior to that. As I mentioned, the leading protagonists here are of Japanese origin. the two individuals should really get most, if not all, of the credit are Mitinori Saito, who was and remains at Kyoto University, and Katsuhiko Hayashi, who used to work with Dr. Saito, perhaps as a postdoc, but who is now on his own at Kyushu University and making independent contributions to this field that you could say were unanticipated and definitely increases the productivity of the Japanese contingent. The experimental paradigms that have been deployed here are, as you can imagine, those of the rodent, and increasingly an effort to apply some of this to the human, although I can state at this point that the human circumstance is still far from ready for prime time and will need much more work, let alone approval by the appropriate authorities before it is applied. But that should not prevent us from looking at the future and anticipating what is likely coming our way. All of this goes back to another central concept, and that is the concept of pluripotent stem cells. These cells come in two varieties, embryonic or induced. The power of stem cells, of course, is that they can become any other cell type. We could guide these cells to become amacrine neurons of the retina, which some patients lack. We could guide these cells to become dopaminergic neurons and maybe implant them in the striatum in patients who are suffering from Parkinson's disease. Or we could make motor neurons, just as an example, and potentially cure spinal paralysis. So the possibilities are endless, and we are today just focusing on the reproductive implications, and specifically about the derivation of gametes from stem cells and originally really from any somatic cell in the body. Science magazine understood the significance of all of this and dedicated a major spread to this topic in 2021. The New England Journal of Medicine, not wanting to fall behind, did some of the same in 2022. And even more importantly, last year, the National Academy of Medicine dedicated a 2.5-day workshop, which I chaired, which involved the latest and greatest in the field of in vitro gametogenesis. This will likely be followed by a report of a consensus committee to be convened by the National Academy of Medicine, but that has not happened yet. It is also instructive to take a look at the annual report of the Nuffield Council on Bioethics, which is the premier British ethics organization, which looks at a variety of issues from an ethical point of view. But it's interesting to note that in the arena of human reproduction, in vitro-derived gametes tops the list, to be followed by time limits on maintaining embryos in culture, womb replacements, preconception screening, whole genome sequencing of embryos and fetuses, genome editing of gametes and embryos, and egg freezing. But first amongst those was in vitro-derived gametes. So in vitro gametogenesis, we can all agree, is a disruptive technology. Whether or not it's achievable in the human still remains to be seen. And its timeline is also somewhat uncertain, but I think it's fair to say that it will happen sometime in the lifetime of the young members of this audience. Exactly when, of course, we don't know, but I think we are on the precipice of this becoming a reality. One cannot help but recall the 1932 book of Aldous Huxley, Brave New World, wherein he made several predictions about the future of reproduction. He suggested that eggs will be created via ex vivo ovarian cultures, that embryos will be created via in vitro fertilization, and that newborns will be generated via ex vivo ectogenesis. Now, we know, of course, that he was spot on when it comes to the prediction of in vitro fertilization. The notion of producing eggs via ex vivo ovarian culture may have to be modified in light of in vitro gametogenesis. And as for ectogenesis, it's another futuristic topic. The notion that women will be freed up, if you want to call it that, of the burden of gestation, and that gestation will actually occur ex utero in an artificial uterus. That is being pursued. That's nowhere near realization, and of course, deserves a whole separate discussion. In chapter 1 of Brave New World, it is stated by the director of so-called hatcheries and conditioning, one egg, one embryo, one adult, normality, making 96 human beings grow where only one grew before progress. And in some respects, that is not far from the truth, because we can now readily create 96 well plates, which are all populated by in vitro derived gametes. So let's begin by talking a little bit about the early obstacles to the realization of this concept. The main theoretical obstacle was a theory that prevailed in the last century. It was known as the Soma germline barrier hypothesis, and it was promulgated by August Weismann, a professor of zoology at the University of Freiburg, who in his book, The Germplasm, A Theory of Heredity, argued that while the germline gives rise to the soma, which renders the germline, if you will, immortal, the soma, for its part, never gives rise to the germline, which is why we can refer to it as mortal. In hindsight, this was wrong, if we can use that term. Of course, the theory was perfectly appropriate for its time, But with in vitro gametogenesis, this conclusion is probably rendered, in hindsight, null and void. We can, courtesy of pluripotent stem cell formation technologies, create eggs from virtually, and sperm from any tissue in the body. But before we go there, we need to review briefly the notion of somatic cell nuclear transfer, or SCNT for short, which is important for the full appreciation of in vitro gametogenesis. It was early on in the 30s that John Garden, PhD, a British biologist, published a paper in Nature titled Sexually Mature Individuals of Xenopus laevis from the Transplantation of Single Somatic Nuclei. He is really the father of so-called somatic cell nuclear transfer, which means taking a nucleus of a somatic cell, injecting it into an enucleated oocyte, and creating a quasi-zygote, which then develops into a blastocyst, which gives rise to an inner cell mass and eventually to a human being. You can also derive embryonic stem cells from the blastocyst, which is the conventional way of doing so. More recently, in 2006, Shinya Yamanaka from Kyoto University published a paper in Cell titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. This was an alternative approach to the creation of embryonic stem cells. He also started with somatic cells, but then transfected them with select transcription factors and gave rise to what is known as induced pluripotent stem cells. But pluripotent stem cells, nevertheless, for all practical purposes, which means that these are cells that can go anywhere, meaning they can become any cell type in the body. It is of note that both individuals were the recipient of the Nobel Prize in 2012 for very good reasons. And that discovery is really what undergirds in vitro gametogenesis, which involves a somatic cell, let's say a skin cell, that using one of those techniques, we convert either to an embryonic or induced pluripotent stem cells. And then if we know the recipe, we can then guide that pluripotent stem cell towards a gametogenic fate. Question is, what are the prerequisites for making this happen? Well, you obviously need to know the cues that need to be applied to these stem cells, the sequence by which they need to be applied, the timing of their application, and the environment wherein they are encased, or the so-called niche. I don't have to tell this audience that oocytes are surrounded by granulosa cells, and sperm are surrounded by Sertoli cells. And that's not a coincidence. Those somatic cells play a role in the evolution and maturation of the gametes. And so if you're going to produce gametes in vitro, you are going to have to simulate the in vivo process as well as you can. You could say that the beginning of this field, perhaps, dates back to 2003, at a point in time when Hans Schöller, who is now director of a Max Planck Institute in Germany, was a postdoc at the University of Pennsylvania in Philadelphia. Published a paper in Science titled Derivation of Oocytes from Mouse Embryonic Stem Cells. This is perhaps the very first report of this possibility. An example of the oocyte in question is shown in the right lower hand side of the slide. That same year, Japanese investigators reported more of the same on the pages of the Proceedings of the National Academy of Sciences, a US-based publication of great significance. Also, a year later, on the pages of Nature, George Daly, who is currently the dean of Harvard Medical School, published a paper titled Derivation of Embryonic Germ Cells and Male Gametes from Embryonic Stem Cells. And Rene Rejo-Pera, who at the time was at UCSF, published a paper in Human Molecular Genetics on the same subject. So relatively early on, inklings of what was possible were beginning to emerge. In the next 5 to 10 years, a series of high-impact papers on this phenomenon were published in a variety of journals. This happens to be relevant to rodent stem cells. But then a comparable series of high-impact papers was published involving human germ cells that were created in vitro. And so if you look at the state of the art in 2015, we could say that the germ cell specification pathway is not conserved between the rodent and the human. The two are different. You can learn from one, but you cannot simply copy the rodent process and expect it to work in the human. And we could also say that the genetic networks involved are quite distinct as well, all of which was meant to say that it's not enough to study the rodent. We have to study the human if we want to make this happen in a human being. So to summarize where we were in 2015, we could take embryonic stem cells or induced pluripotent stem cells and convert them to primordial germ cell-like cells, which is the process in the body would occur extragonadally before the gonad is ever involved. What we couldn't do at the time is take these primordial germ cell-like cells and convert them into mature haploid post-meiotic gametes, a process which takes place in the ovary or the testis proper. So in 2015, we were still a long way away from making in vitro gametogenesis work. The extragonadal piece was working, the intragonadal piece was lacking. And so the final frontier of in vitro gametogenesis, quote unquote, all the way, still had to be reached. Enter Katsuhiko Hayashi, whom you met earlier, who is now at Kyushu University, who published a paper in Nature in 2016 titled Reconstitution in Vitro of the Entire Cycle of the Mouse Female Germline. So for the first time, he was able to take a stem cell and make a female germ cell, albeit in the rodent. He started with stem cells, converted them to primordial germ cell-like cells, combined them with gonadal somatic cells, which gave rise to primary oocytes, eventually to M2 oocytes, which upon fertilization gave rise to an embryo, a blastocyst, which upon gestation gave rise to living pups. And so in the rodent, at least, we're now in a position to say that we could start with somatic cells, convert them to stem cells, convert those to germ cells, in this case oocytes, fertilize those, and actually give rise to a pregnancy and delivery. He also generated ovarian follicles or granulosa cells in the process, but that's a secondary issue. More of the same transpired on the male side, the protagonist here being Mitinori Saito. In a paper in Cell Stem Cell titled In Vitro Reconstitution of the Whole Male Germ Cell Development from Mouse Pluripotent Stem Cells, Mitinori Saito did more or less the same, but in the male. And when I say the male, of course, the rodent male. What was still lacking, and is still lacking, is the human paradigm. There were some efforts to get there, and perhaps you could say that we are somewhere halfway or along the way, but we have yet to reach the in vitro gametogenesis promised land, so to speak. Science magazine in 2018 published a paper by Mitinori Saito, wherein he started the process with human primordial germ cell-like cells, combined them with somatic cells, and matured them all the way to the oogonial stage. But he was unable to reach the point of meiotic initiation, which is, of course, essential if fertilization is to be accomplished and conception is to follow. More or less at the same time, Kotaro Sasaki, who was at the time a faculty at the University of Pennsylvania, published in Nature Communications a paper titled Reconstitution of Prospermatogonial Specification in Vitro from Human-Induced Pluripotent Stem Cells. In other words, he produced the male counterpart to what Mitinori Saito was seeking to achieve on the female side. So when it comes to human in vitro gametogenesis, we are still lacking the production of M2 oocytes or full-fledged spermatozoa. That, of course, will be essential if fertilization is to be accomplished. So as we sit here today, we have to, first of all, acknowledge that this is an evolving field. We have yet to reach the endpoint, so to speak. But there is definitely a scientific and translational promise here, which can't be too far away, but of course, it's impossible to predict how long it will take for this technology to be applicable in the human and eventually approved in the human and eventually applied in the human. We cannot make those predictions except to say that those are future developments. But when it happens, This will give us an inexhaustible supply of germ cells. It will allow us to do what is known as functional non-mutant analysis, which is to say, knock out one gene at a time and ask, what is the function of that gene? It will allow us to study the heretofore unstudied germ somatic cell interactions. And from a practical point of view, it would allow us to reverse germ cell failure, that is to say, infertility. We could replace missing or defective gametes, potentially. We could gene edit those gametes if that was desirable or acceptable or acceptable at the time. And amazingly, and I will touch upon this a little more later, it will allow, for the first time in human history, for same-sex parenthood, genetic parenthood. That is to say, two men could, given this technology, in principle, that's a separate conversation, a separate lecture, in principle, could be the genetic parents of a child. Of course, they will need a surrogate to carry the pregnancy, but they could otherwise be parents for all practical purposes. And the same would be true for two women who live together and wish to be the genetic parents of a child. And so same-sex relationship in the future is not going to be a barrier to genetic parenthood. This will also have an impact on IVF. Right now, as you know, to secure eggs, we need to stimulate the women in question with gonadotropins, and at the appropriate time, engage in transvaginal egg retrieval. That may not be necessary. We also may not need gamete donation, which would involve the same process today, but could be simplified by starting, for example, with a skin cell. It is also likely to convert IVF to a laboratory procedure because the in vivo part, that is to say, stimulating women to produce eggs, retrieving eggs from women, that could be bypassed. And the rest of it is primarily an in vitro procedure. The only time you revert back to the in vivo state is when you replace the resultant blastocyst into the womb of the recipient woman. We may still require endometrial preparation and, of course, transfer, but on the whole, this will definitely change, and you could probably say simplify IVF, and in theory could improve affordability and access. I say this because IVF is still rather expensive. Not every state has an IVF mandate. Access is not always so simple. But at the same time, being realistic, I would say we've been there before. And while this would make sense in some way, in reality, the price often fails to decline, which is what one might hope for. Almost going without saying, this could not be applied willy-nilly. It will require regulation. And in the United States, if in vitro gametogenesis were to become a reality, it will definitely require FDA jurisdiction. The office that handles this type of approval is known as CBER, or the Center for Biologics Evaluation and Research. And one would really have to get their blessings before this is clinically applied. This procedure will fall under a category that CBER abbreviates HCTPs, or human cells, tissues, and products. So this requires a formal application to the FDA. On the part of the FDA, it requires the convening of an expert committee that will consider the application and then vote up or down on its approval or disapproval. And so all of this is, of course, down the line, but it's important to keep in mind as we speak about all of this today. Which brings us full circle to the Nobel Council on Bioethics and the fact that it places in vitro derived gametes at the top of its agenda. Not perhaps because it's likely to be the next breakthrough in the sequence, but perhaps because it may be the most important next breakthrough in the sequence, although, as we said, there are many others, potential breakthroughs out there, like womb replacements, that deserve their own treatment and are being worked on, but are clearly much further away from realization when compared to in vitro gametogenesis. So the notion of gametes to order is not all that far away now that we have acquainted ourselves with this topic. And all I can say to you and to myself is stay tuned. This is a dynamic, fluid field that can surprise us anytime. Japanese investigators primarily, although there are a few American groups now that have entered the fray, but the lead, one would have to say, is still from Japan. And we don't know specifically what they're working on, how far they've gotten, and what's the next breakthrough. But this track record that I shared with you today of the Japanese investigators who brought us this far argues strongly that more of the same is in store. It's just a matter of time. So stay tuned, and next time when we reconvene, hopefully there'll be more to say about this field, which remains a dynamic field in motion with very promising applications that has yet to be fully realized. So thank you very much for the opportunity to share with you the latest. I trust you will stay on top of it as new developments evolve. This is definitely something the residents in the room are likely to witness in their professional lifetime. And so it's worth knowing about, and it's worth paying attention to. So thank you very much again for the opportunity to share this morning with you. Thank you, Jack.
No, thank you, Lily. I think we have a few minutes for some questions, so I don't know if anyone has a specific question. I have one, Ellie, and
Sure.
this is so exciting. But when the Japanese tried to do this in the human and on their papers, what was the source for their stem cells? Does he make us a difference, the source?
No, I mean, they use skin because it's simple. Although I would argue that the buccal cells are even simpler. For one reason or another, nobody seems to go that way. But as you noticed, I think I titled my lecture
Yeah,
because to me, I mean, that's as simple as a swab. Skin cells are accessible, of course, but it does involve, a local anesthetic, probably, and a small biopsy. But for one reason or another, buccal cells have not been deployed as starting material. So skin cells, probably simply because of accessibility, have served as starting materials here.
any other questions?
I should also say that there have been some work also by Japanese investigators using the primate because there are obviously less limitations on certain aspects of the work. Not much is written about it, but I would think that that's a wise strategy because certain parts of this experiment are best tested in primates before they are applied to humans. They have done some work, but for one reason or another, we haven't seen too much in the way of published work. I should say, which I meant to earlier, that when I first became aware of this work, I used a trip to Japan in 2012 or 2013 to book a flight to Kyoto and visit these two gentlemen and spend half a day with them long before we got to the point we are at right now. Although I saw them again virtually anyway last year at the meeting of the National Academy of Medicine. But Kyoto, you should know, is of course leading high quality university with Nobel Prize winners. They also have a stem cell institute, which makes the work in question amenable and also logical. And so for us who are understandably American-centric, we tend to lose sight sometimes of the power that resides elsewhere. And in this case, there is very little question but that if a Nobel Prize is ever to be awarded for in vitro gametogenesis, and I think it should, my guess is that Mitinori Saito and Katsuhiko Hayashi will be leading candidates, which is only appropriate.
Wow. Well, thank you so much for that incredible insight and bringing to our attention these incredible advances in science. It's really exciting. We're really looking forward to the next chapter. But I don't know if anyone has any other questions for Dr. Adachi. I think, Ellie, this is at a totally different level. So
No, I mean, it's really in final analysis. This is, day-to-day clinical care issue, but to us, it's still science fiction. To our successors, it will be,
Reality, yeah.
yeah, I mean, will be just a daily affair. Um,
Anyway, I really want to thank you. I really want to thank you through the years for your support to Temple and this department. I'm gonna just tell Enrique how happy we were
Thank
about having
you.
you as an invited guest, and I'm really looking forward to keep learning and hearing from you and your almost every 2 weeks email about a new paper because you're really a source of inspiration.
Well, I'll reach out when I'm in Philadelphia and maybe we can
Yeah,
have
you bet.
coffee. Yeah.
You bet. You bet. Thank
Okay.
you again. Thank you, really.
Thank you.
Bye bye.
Bye-bye.
All the best.
Thank you.